Block. I did my thesis at GMIT on the reproduction of the short-beaked common dolphin. So the main aim of my thesis was to describe the developmental and seasonal changes in testicular morphology of the northeastern Atlantic short-beaked common dolphin. A second aim of ours was to develop a classification system specifically for the species in order to determine periods of high and low reproductive activity. Our initial classification system was based on the criteria of spermatogenetic activity. And an additional aim we had was to investigate the health impact on the reproduction. But first, I'll give you an overview of the species. So the short-beaked common dolphin has a broad geographical range, but we focused mainly on the northeastern Atlantic population. Over there, it's the most abundant species, and it's protected by the Azkabans. The species has known seasonal migration inshore and offshore based on prey movements, which are in turn based on environmental conditions. The species is a top predator, which means it plays an important role for maintaining ecosystem balance. Most of my research was based on histological examination of testes. So the samples were obtained from bycatch and stranding programs organized over 30 years along the northeastern Atlantic coast. During necropsy, testes weights, blubber depth, and other body size parameters like length, etc. were measured. And the individuals were all aged based on their teeth. These two steps were done prior to the start of my thesis, and I was mainly involved from step 3 onwards. However, a lot of the slides, histological slides, were also made prior to the start of the thesis. So most of my research was based on, I spent doing histological examination through camera systems of morphometric parameters. In order to explain those parameters, I will give you a recap of testes morphology. So this is where the testes are located in a dolphin, and they can be split up in the epididymis and the seminiferous tubules. Within the tubules, spermatogenesis happens. This is also called sperm production, and there are 5 main stages, starting with spermatogonia, then primary spermatocytes, secondary spermatocytes, spermatids, who then mature into mature sperm or spermatozoa. As I mentioned, so we used parameters. The 9 main parameters are the following. The first one is combined testis weights of the right and left side. Then it's average seminiferous tubule per individual. So we measured the average tubule diameter of the shortest diameter of the roundest tubules. And then we also made an estimation of the amount, and we noted down the presence of the 5 main stages of spermatogenesis per individual. We also calculated the proportion of tubules with sperm, so this one has sperm, and these two do not. We also made an estimation of the shape based on the angularity scale described for particulates. You can see this individual had round tubules, this one had slightly angular, and this one had angular. Similarly, we made an estimation of the surface area of interstitial tissue compared to tubule surface area. You can see this one had a lot of interstitial tissue, but very little in tubule surface area, while this one had the opposite. We also noted down the presence of sperm in the epididymis, but this was mainly used for maturity characterization. And finally, while it's not a testicular morphology parameter, the blubber depth parameter was an important one for seasonal changes. Then, prior to our analysis of our results, we classified each individual based on 2 classification systems. The first system was based on maturity, which was based on the presence of specific spermatogenesis stages, and other criteria like the presence of lumen, as described by my supervisor in her previous work. We had 3 classes: immature, pubescent, and matures. Sexually matures were then further classified into spermatogenetic activity. This is basically based on the proportion of testes that were capable or were actively producing sperm, so had the capability of spermatogenesis, i.e. Were producing viable gametes for genetic reproduction. We described 3 spermatogenetic activity classes, low, medium, and high. Low individuals had a portion of their testes inactive, so a portion of their tubules were not producing sperm. Then the mediums, all of their tubules were in a stage of spermatogenesis, but not all had reached the final stage. And the high actives, all tubules had spermatozoa and were producing sperm. You'll find in literature that there is often a fully seasonally regressed class described, but because our species don't, we didn't use a classification for fully inactives because there weren't any. On to our first results of our main aim, the developmental changes. Sadly, for pubescents, we only had 9 individuals, so we mostly had to compare immatures and matures statistically. You can see that pubescents are a nice in between. Immatures were statistically significantly had smaller testes, smaller tubule diameter, much more interstitial tissue, had by their criteria also only spermatogonia and Sertoli cells, while matures had all stages present, including the sperm. You can see those here. Matures were also much more angular in tubule shape, but you'll see that with seasonality, as this is a summer individual. The species becomes sexually mature at an average age of 11.62 years and an average length of 206.49 centimeters. These were calculated respectively using the sum of fraction of immatures method and a modification of this method. And then onwards to the second part of our main aim, seasonal changes. These are the 4 main testicular morphology parameters, and you can see for all that there is a seasonal trend and a seasonal peak or valley in summer. On the left side, you'll see that tubules start off in winter as small and very angular and become then much rounder and larger. Here on the graph, lower means rounder. Similarly, interstitial tissue decreases in compared to tubule surface area in summer, and in summer also the combined testes weights increase very rapidly, as was also seen in the population on the western coast. Then seasonal changes in sperm production. You saw in the other parameters that the peaks were mostly from June until September, and you see that here too. Here almost all individuals have all of their tubules filled with sperm, and outside of this period you have very strong individual variation. Then you see a similar pattern of this peak in summer from around June until September in the estimated amount of sperm. And interestingly enough, when we look at the seasonal changes in blubber depth, we see that October is the lowest point, and this falls right outside this peak of sperm production and this strong increase or this strong change in testicular morphology. And you see that in this period there is also a nice slope decrease in blubber depth. So this leads us to believe or think that there is likely a lot of energy consumption stored of energy in the blubber, and because reproduction costs a lot of energy, that is why we see this decrease until right after their peak. And then back on to our second aim. So we wanted to classify periods based on activity. So you see here in winter we have most of the population being being low active, and in summer almost all of them are high active, and in between you have medium, so you see a nice succession. We similarly see this when then we add other parameters. So in winter they're mostly low active, and they start to increase in values towards medium active, and then do the peak when they are high active. But interestingly enough, you can see that low active still remain higher in winter, but they do differ between the seasons. And interestingly enough, there is still a large proportion of the sample that is high active in winter. So we know that the species produces sperm all year round, but we hadn't expected that they would be high active in winter. So we further wanted to analyze this. So between summer and winter, we found that there are seasonal changes in testicular morphology. So are there seasonal changes within the high active group as well for morphology? And yes, we do find morphology. So it's not the best classification system based on just one criteria. But interestingly enough, we didn't find a significant difference in amount of sperm. So the proportion and the amount of sperm does not change for the high actives and between the seasons. But then we also, because the classification is based on just that criteria of proportion of tubules with sperm, we wanted to see, well, is there a significant difference in morphology between these 3 activity classes in winter? And yes, we do find that both morphology and the amount of sperm significantly differ between the activity classes, which is interesting. And then while we didn't test tubule shape, you can still see that graphically they are lower, or they are more rounder, the high actives, compared to the other 2 classes. You similarly see this on the photos. So basically we found that there was high individual variation in seasonal regression. You have those that regress towards low, and they change very strongly in morphology and also in sperm production towards low activity. activity in winter, and then you have those that remain high active even during winter, and that and these ones have only change in morphology and not sperm. So then the question is, why do some remain more active in winter? As I mentioned, we had other parameters like body size and health parameters, but we could not find a significant difference in winter between these three activity classes. So we think, okay, it must be other factors that we didn't take into consideration. We have three hypotheses for these. First one is food availability. So we did test blubber depth, and this was not significantly different between the three activity classes. So it might be that just within the blubber there is just a specific lipid content that's different, and so that those that are high active just have more energy. Similarly, there was a study by Keller et al. That found that testosterone concentrations followed a similar pattern as ours in morphology and sperm production. So it might be, and because also that sperm production is dependent on the testosterone concentration, it might be that those that are high active just have more testosterone and aren't decreasing enough in testosterone concentration in winter to fully change the sperm production as well, besides also their morphology. And then lastly, the species is a sperm competitor. So it might be that those that are high active and are able to maintain high activity during winter are just stronger in sperm competition, and it might be that there is a sort of selection towards this. Now, back to our last aim, the effect of illness on testicular morphology. Because we had very little cause of death data and because most of the analysis done were not statistically interpretable, this needs to be further researched. The only parameter we could interpret was the blubber depth, and those that died to infectious illnesses had lower blubber depth compared to the other ones. Here you can see an individual that has ill structures, but because this one did not have a cause of death class, we weren't able to label this as ill. So we recommend consulting a veterinarian who is licensed in dolphins and who can further tell you the effects. And then here you can see, while we couldn't statistically interpret it, graphically you see that there is likely to be no difference in the amount of cells per spermatogenesis stage. And then for our conclusion, so our main aim was completed. We found developmental changes and seasonal changes in testicular morphology. Based on these seasonal changes and based on our initial classification system, we can kind of describe a period of high activity from June until September. While the species was known to produce sperm year-round, we did not expect high sperm production in winter, and we had our three proposed hypotheses for these. And then an important take-home message is that the species has an important ecosystem role as top predator, which means that it's important for conservation. Understanding life history parameters like ASM, LSM, and the breeding season will help in conservation efforts and for population modeling. And then for future research, we can recommend looking deeper into winter actives and also the effect of specific illnesses or just illnesses in general on morphology and on the reproductive behavior. So long, and thanks for all the fish. And here are my references if you would like them.